After the sample is obtained, the loop is presented to the loop check to ensure accuracy of plating. The sample is inoculated onto the plate using one of a library of classic microbiology streak patterns honed by years of expertise in reading plates. WASP can also accommodate customized streak patterns. WASP has an optional dual streaker, which can plant and streak both sides of a biplate at once, maximizing throughput. Depending on sample protocol, the WASP can inoculate enrichment broths or apply susceptibility discs at the warehouse carousel module. The gram slide prep module automatically prepares a gram slide and inkjets patient data directly onto the glass slide, eliminating the need for label. After processing, samples and plates are offloaded from the front of the instrument. The entire WASP system uses a targeted HEPA filtration system, focusing on high-risk areas where specimens are open and optimizing specimen containment and confinement. By maintaining a close relationship with the microbiology community, Copan continues to make innovative strides and has designed WASP as an open platform modular instrument for the seamless addition of new features and capabilities.